Hello. Good morning, ladies and gentlemen. We are about to start our award ceremony session today. My name is Bernard Marty. I am president of the European Association of Geochemistry. One important task for our society is the recognition of scientific excellence at all stages of a career. Therefore, as president of the EAG, European Association of Geochemistry, it's an honor to present the 2018 EAG Award. And just before starting, I would like to sincerely thank all those who submitted nomination and to all the members of the award committees. Nominations for the 2019 awards are now open, and I encourage you to recognize the serving colleagues by submitting your nomination. Our first award is the Friedrich Utermans Award, recognizing exceptional contribution made to geochemistry by the younger generation. This year, I am honored to present this award to Morgan Schaller from Rensselaer Polytechnic Institute. Morgan is best known for documenting major changes in atmospheric CO2 through 32 million years of the early Mesozoic, including those associated with basalt emplacement during the Triassic, Jurassic, and subsequent weathering. And most recently, the discovery of impact spherules at the Paleocene-Eocene boundary that was reported in Science. Morgan, in recognition of your exceptional achievement, I am honored to present you with the 2018 Utermans Award. Please, Morgan. Our second award is the Science Innovation Award, recognizing mid-career scientists. This year, it is named in honor of Nicholas Shackleton for his work in climatology. I am delighted to present this award to Jess Atkins from the California Institute of Technology. Jess has pioneered the field of deep-sea coral paleoceanography, which has helped us understand the role of the deep ocean in climate change and abrupt climate variations. Jess, in recognition of your innovative work, let me present you with the Nicholas Shackleton Medal. And our last award is the Urey Award, recognizing outstanding contribution advancing geochemistry over an entire career. This year, this award goes to Sue Brantley from Penn State University. Sue has developed an exceptional insight of the physico-chemical investigation of the Earth's surface. She has ventured from her background in low-temperature aqueous geochemical kinetics into numerous other disciplines, including geomorphology, microbiology, and geophysics. Sue, in recognition of your remarkable achievement, let me present you with the 2018 Harold Urey Medal. Now I am going to call Roberta Rudnick, President of the Geochemical Society, who will introduce today's plenary speaker.
Thank you, Bernard. Good morning, and welcome to our second plenary session. Together with Bernard, I'm very pleased to present an award bestowed jointly by the Geochemical Society and the European Association of Geochemistry, the Paul W. Gast Lectureship. This honor recognizes outstanding contributions to geochemistry by a mid-career scientist. Paul Werner Gast was the first V.M. Goldschmidt medalist in 1972 and pioneered the study of rare earth elements. This year, we are delighted to present the award to Dr. Caroline Slomp, Professor of Marine Biogeochemistry in the Department of Earth Sciences at Utrecht University in the Netherlands. Her innovative work touches on sediment biogeochemistry, paleooceanography, reactive transport modeling, and submarine groundwater discharge. In her lecture today, she will address oxygen loss in coastal waters and its impact on geochemical cycles. But first, please join me in congratulating her on receiving the 2018 Paul Gast Award. And now I would like to welcome Shuhei Ono, last year's Gast Award recipient, to come forward and formally introduce this year's lecture. Shuhei?
Okay, thank you, Roberta. I'm pleased to introduce Professor Slomp to deliver this year's Gast lecture. So her research focuses on improving the quantitative understanding of the cycling of elements that are important to life in marine environment. Okay? In her research group, field and laboratory work are typically integrated with modeling. The central theme is the assessment of the response of ancient and modern environments to perturbation. Currently, low oxygen marine systems such as present-day Baltic Sea, Black Sea, and Arabian Sea, and the Atlantic Ocean in the Cretaceous are the key focus areas of her group. So please join me in welcoming her to Goldschmidt 2018 Gast lecture.
Thank you, Shuhei, for the very nice introduction. I'm very, very honored to give this lecture here today, and yeah, it's a great pleasure. What I want to talk about is a topic that has been of interest to me these last 10 years and started with a field trip to the Baltic Sea. And since then, I've done a lot of additional field trips to anoxic areas, and I want to bring across two things here. One is they have a fascinating geochemistry, but the other thing is that large-scale ocean anoxia has devastating effects on environments, and so we also need to use our geochemical understanding to do something about it, to get rid of that anoxia. And so those are two things I want to address today in my talk. But let me give you some more context first. This figure shows the most recent overview of coastal areas along all coasts, along all continents, suffering from low oxygen conditions. And these are called hypoxic areas where oxygen concentrations are lower than 2 milligrams per liter in the deeper waters. And in some of these environments, there's also sulfide in the bottom water, and that's extremely toxic to life. So that especially is an environmental disruptor. Now, we know why these coastal environments are anoxic. What we're seeing is that nutrients are leaking from land, nitrogen and phosphorus mainly, and are eutrophying coastal waters, are leading to growth of algae. These algae sink to the bottom, to the seafloor, start degrading and remove the oxygen. So we see an enhancement of the oxygen uptake. And if you then look at the principal terms in an oxygen budget for a coastal zone, as shown in this very nice figure, you can see that you have oxygen demand occurring in these bottom waters, but also from the sea floor. And you also see oxygen supply, and that oxygen is supplied through physical transport, either vertical or or lateral, and also from the atmosphere through air-sea exchange. And there the solubility of oxygen and the temperature dependence of that plays a role. So here we see two things. We see oxygen demand dropping, and the other thing is that oxygen supply can change. And global warming is one of the key drivers right now of the change in oxygen supply. So through the solubility effect, but also by affecting circulation. Now, we know that this is all a relatively recent phenomenon. Well, it depends how old you are, but anyhow, 1950, that's not that long ago. And what we're seeing is that before that, there was not that much eutrophication. And after 1950, in concert with or due to the change in human population and the use of fertilizer, a lot of rivers and land and aquatic systems on land have become eutrophic, and also the coastal zone then followed. And what we see is a timeline from developed countries being most eutrophic first, and showing these symptoms early on, and developing countries now following. Now, one very important thing that I want to talk about today is the timeline of these processes. So what we see is we have this eutrophication occurring, we have hypoxia occurring, but then the question is, how will these systems develop further? And will they recover? Now, there's been a lot of research on that, and I can't summarize all that. So I want to do this in a very, very simple way. And I'm going to show you essentially two extreme possibilities. One is the extreme case that you add nutrients and that your system becomes low in oxygen and maybe even sulfidic, but that you essentially can just retrace your steps and go back to the initial condition. That's great when you make a mistake and you can just simply go back. Wish that would always happen. But what you see is that in coastal environments that is possible, but it only holds for environments that are relatively shallow and that have relatively short residence times. And many, many systems are not like that. And they have very different trajectories of responses to changes in nutrient load. And a range of possibilities is actually summarized here. So again, you see the increase of nutrient load and then the response of hypoxia here. And what you can see here is what happens in much larger systems. What you see is that an increase in nutrients may not directly result in hypoxia. There can be quite a delay in the onset. What you also see is that you might suddenly pass a threshold and then get into deep trouble without a big change in load. And then you also see the hysteresis in the response, that when you decrease the load, that you might not follow the same trajectory back, and that you might end up at a different baseline, even though the load is the same as what you had in the past. Now, we know all this, and we also know a lot of the causes. We know that climate plays a role, that solubility of oxygen, again, as I mentioned, global warming plays a role, and can explain why you don't go back to the same state. Another important factor is the biological side of things. So you can have other primary producers suddenly taking over, or other animals, macrofauna in the sediment playing a role here. Now, because this is a geochemical conference, I decided today to not talk about all that biology, but essentially talk mainly about the geochemistry, and especially the microbially driven biogeochemistry that plays a role in these types of nonlinear responses. And what really matters there is the shift in microbial pathways of organic matter degradation upon oxygen loss from seawater. And what you get is, when you have less oxygen, you essentially get a shift in pathways to other electron acceptors and other processes where, especially in the context of this talk, iron oxide reduction, sulfate reduction, and methanogenesis are very important. And what you then get in a system like that is that reduced products of these reactions become available in the seafloor, but also start seeping out into the overlying water and can be released to that overlying water and can start using up oxygen. And so we get an excess oxygen demand because of all that, those reduced substances coming up. And especially the longer low oxygen occurs, the more products can come up and lead to nonlinearity in the system. Another factor that's important is the release of nutrients. So phosphate and ammonium can be released from organic matter and can also escape out of the sediment and fuel more algal blooms, leading to more oxygen loss ultimately when that organic matter decays. Okay, so these are the things I'm going to talk about today, and I'm going to focus on just, again, a part of this, and that's essentially I'm going to talk about 3 examples of microbially driven processes that can affect this trajectory of hypoxia, both the onset and recovery. And we're going to look at the key role for iron oxides as a buffer for hydrogen sulfide, because iron oxides can take up hydrogen sulfide and make sure it doesn't escape to the overlying water. And then the other thing is that it can remove phosphate, which is an important nutrient. Now for something really new, and that is that what we have realized is that the type of bacteria that you find in sediments can actually contribute to nonlinearities in the response. And bacteria that there's a type of new bacteria that have recently been discovered, so-called cable bacteria. And these actually do something that no one thought could be possible. What they do is they're centimeters long, they're multicellular, but one organism can actually couple the oxidation of sulfide with the reduction of oxygen and transporting electrons along their body. And I'm not going to go into all the detail of how these bacteria do that, but I do want to talk about the major effect that they have on the sediment geochemistry. And what they do what they generate when they do this process is H+. And this leads to very acidic pore waters. And so on the right-hand side on this figure, on the slide, you can see that the pH in the pore waters can become very, very low. And so sometimes below 6.5. Here you see it reaches about 6.5. Now, these bacteria were discovered in lab experiments and in a lab system in Denmark in the group of Lars Peter Nielsen. And in that group, they also figured out what that low acidity did to the chemistry. And what this acidity does, it dissolves iron sulfides and it dissolves iron carbonates, and you can get a lot of dissolved iron then in the pore water that can diffuse down, form iron sulfides again, but also diffuse up, forming iron oxides in the surface. So what they do is they transform the chemistry of your sediment, removing iron sulfides and forming iron oxides. Now, this is very, very important from an environmental point of view because, as I just mentioned, iron oxides can buffer sulfides and bind phosphate. But okay, first, as I said, first of all these data came from core incubations in the lab. This was a discovery that was based on a core that had just been sitting in the lab and someone suddenly wanted to use for an experiment. And so the big question that then came forward was, well, how relevant are these bacteria in coastal systems? And do they matter, especially in the context of this lecture, for these seasonally hypoxic coastal systems, so systems where you have low oxygen part of the year? So we set out to study this in a Dutch estuary that was dammed on 2 sides and it's turned into a lake, but it's completely marine. And this system is seasonally low in oxygen and has high oxygen part of the year. And the striking thing about, one of the very, very striking things about this system was that even if you look at all the water column, if you look at all the water column records for the system over the past 40 years, because that's already how long it has been dammed, you never see hydrogen sulfide appearing in the bottom water. And as I mentioned, hydrogen sulfide, that's the toxic compound. So what is happening here? What we did was then look at if these cable bacteria were indeed present. And what you can see here is the seasonal variation in the occurrence of different organisms that are relevant here, and one is cable bacteria, and indeed they were there in spring. And they were, after spring, microfauna that mixed the sediment came in, then the sediment became anoxic, and then another sulfur oxidizing species took over. But as I said, the focus here is on these cable bacteria and what they can do to your system and ultimately how they affect the development of low oxygen. So if you find an organism, it doesn't mean it's doing something, and it doesn't mean that it's really important. And so the next step was, of course, to see if we saw this low pH, and if this Campylobacteria was really affecting the chemistry and was doing something. And indeed, what we saw in spring in this area was indeed very strong activity of these Campylobacteria, low pH, very strong release of dissolved iron, calcium, and sulfate to the pore water. And this is really unusual because calcium, well, typically you would say it should act conservatively in a system like this, but no, here you see very strong dissolution of that. And the iron concentrations are also extremely high. So what we're seeing in this system, we're seeing a microbe that is actually strongly affecting and determining the geochemistry. Now, then the question, of course, is, are they, as already mentioned, you expect that these bacteria are then, they might, we saw that in the lab experiments, are they affecting this iron chemistry? Now, we looked at that over the whole season, but I don't want to bore you with all those pictures, so I'm just showing the contrast here from the period when the Campylobacteria are there and when they disappear. And what you can see here is that when they're there, there's no iron sulfide in the surface sediment, and there is a lot of iron oxide and also phosphorus associated with that in the surface sediment. So what we're seeing is that in this environment, in this coastal lake environment, iron sulfides are being removed by bacteria and are being replaced by iron oxides, and that they are retaining phosphorus. And that red dotted line is the typical concentration that you see in summer and fall. And so this is a pattern that reoccurs every year. I always like showing pictures if they show what the chemistry shows, because what the profiles show. And this figure clearly shows what I was just illustrating with concentration profiles. And it shows a thick layer of iron oxides and below that iron sulfides in the period of Campylobacteria activity. And then in August, very strong iron sulfide-rich sediments. So then those iron oxides are gone again. So what is happening here, and why am I telling you all this detail, giving you all this detail about this geochemistry? Well, what we're seeing in this system is that the fact that you have these bacteria that change the chemistry of your sediment is really important for the sulfide escape to the overlying water. What we see is that in the system, in summer, iron sulfides are transformed to iron oxides, and these iron oxides, when the Campylobacteria die, they stay in the sediment, and when sulfide builds up and more anaerobic degradation occurs in summer, this sulfide does not get out of the sediment, but is retained by these iron oxides. And this was termed a firewall for H2S, so no escape to the water column in summer. And interestingly, other types of bacteria, like the Beggiatoa, that are there typically they're in fall, do not do this. And they are essentially, they don't really affect the iron cycle. So what we're seeing is that the population of microorganisms in the sediment are actually controlling the chemistry here. They're not only controlling the iron chemistry, but also having a big impact on the phosphate chemistry. And what you can see in this figure is the exchange of phosphate from the sediment and between the sediment and the overlying water. And what you can see here is that there's no escape of phosphate from the sediment in spring when Campylobacteria are active. And so in winter, this leads to a big pool of phosphate being retained in the sediment. Okay, so the next point then is, okay, well, we find this in this obscure Dutch lake, man-made, dammed estuary, but what about that bigger picture that I started with? How important is this for hypoxic coastal environments worldwide? And is this buffering, this creation of an iron oxide-rich layer by a type of bacteria, is that actually important at the global scale in these systems? And does that actually hinder the further development of hypoxia in these systems? Well, that's a question we cannot answer yet, but it is a question that we should answer. And before you answer it, you also have to think about what do these types of bacteria need. And we know they need oxygen because they need that for their metabolism. So they need oxygen in bottom water or nitrate. We also know they need sulfide. They can get that from sulfate reduction. But they also can get that from those iron sulfides that they dissolve. So there are two sources there. And they don't like too much sediment disturbance. As I said, they're centimeters long. And so if you start disturbing the sediment a lot, their cables will break. And so you don't want too much bioturbation. And you can see an example of a cable on the right-hand side of the slide here. Okay, so as a first step, we can't go to all environments right now, but as a first step, we went to the Baltic Sea. And the Baltic Sea is the most, or let's say the largest human-induced low oxygen zone or anthropogenically caused low oxygen zone in the world. And so what we did was look at the distribution of cable bacteria there. And on the left-hand side, you can see the oxygen distribution. You see the deeper areas that are completely sulfidic, so below 70 meters, so the bottom waters contain only sulfide there. And then you see the red area where the waters are typically seasonally hypoxic. Before we came there, we had just seen, the system had just undergone a temporary change, and that was due to the fact that there had just been a recent inflow of oxygenated water from the North Sea. That happens about every 10 years. So then the southern part of what we are often called permanently anoxic basin suddenly can get oxygenated. And so we had a range of 4 types of sites here now. We had an oxic site, we had seasonally hypoxic sites, and we had completely anoxic sites, and we had reoxygenated sites. And so the big question was, where would we find the cable bacteria? And where would they have an impact? And would we see the same as you see in the lab experiments and in that Dutch lake? And based on what I was explaining already, you expect them in the seasonally hypoxic areas because they need to connect sulfide, the sulfidic, and the oxygenated zone. So that has to be close together. In a place where you don't have oxygen, you don't expect them to grow. So when we looked at that, what we actually saw was something that fit with that model. So what we see here is the density of cable bacteria in these different areas where the oxic area actually has some cable bacteria, but the anoxic one does not. So you hardly have any cable bacteria. Also in the reoxygenated sites, you don't find them. But you do find them at the seasonally hypoxic sites. And I'm going to concentrate on those mostly. But then the question is, why at those seasonally hypoxic sites? Well, as said, they need oxygen, but they also need sulfide. And so if you just simply look at where sites are with high sulfate reduction, you see a pretty good correlation here between sulfate reduction and the density of these cable bacteria. So what we're seeing is that the presence of these bacteria is determined by how much sulfide there is, but also how frequently there is oxygen. Now, to get back to the system scale impact, so as I mentioned, what we're looking for is a biogeochemical impact. And when we looked at the iron chemistry at these sites, we saw exactly the same as in those lab experiments that they did in Denmark and as we saw in the Dutch lake. And what we see again is a buildup of iron oxides in the surface and a removal of iron sulfide. This is just a snapshot in time after a period where we think these cable bacteria have grown for a while. So the message here is we have sort of ecosystem engineers where bacteria can actually create a layer of iron oxides that can buffer hydrogen sulfide and can make sure it doesn't escape to the overlying water. And this may explain why in some parts of the Baltic Sea, and especially the Gulf of Finland, which is the most eutrophic part of the sea, that the waters there are not anoxic and sulfidic, and that they do go low in oxygen, but you never see sulfide or you don't see it often in the bottom waters. So it seems like these bacteria are playing a role in keeping that sulfide out. Okay, so this was my first example of a microbial process, which is really new, that can impact the trajectory of hypoxia. Because if we are looking at the system where this occurs, if you look at the transition of environments from oxic conditions to anoxic conditions, they usually pass through this period of seasonal hypoxia, of this phase of seasonal hypoxia. And so in this trajectory, the lower trajectory with increased nutrient load, these caelobacteria can hinder the further development of low oxygen in such systems. So it's really important to know more about their functioning and also their occurrence. Now, my second example also relates to iron and sulfur chemistry. And I'm going to talk about the Baltic Sea again. So this is the second example for the Baltic Sea. And it's something that has been puzzling to many researchers, that if you look at the transition of the Baltic Sea, this huge dead zone, into sulfidic conditions, that transition is nonlinear. So what you see is that waters were oxic around 1930, and there's no data between 1930 and 1960, but, or at least not for this site. But then you see 1960, oxygen's a bit lower, but you don't see a lot of change between 1960 and 1980. But we know the system was becoming more eutrophic at this time, so what's going on? And then suddenly, 1980, we see this big switch into sulfidic conditions. So on the y-axis, the sulfide concentrations, they rise when you go in a vertical, if you go down. And so what you see is that between 1980 and 2010, the waters became more and more sulfidic, with sometimes these little blips where the oxygen comes back because of inflow of water from the North Sea. And so, but then again, the hydrogen sulfide comes back quicker each time. So it looks like there's something there that's either creating more hydrogen sulfide or that is actually removing and buffering that hydrogen sulfide. And so again, we thought of different mechanisms. There's some people that have suggested that there's extra zooplankton coming into the system with that North Sea water. But we think that a much more plausible hypothesis is that there's a temporal variation in the incoming iron oxides in the system and a buffering of the hydrogen sulfide. And here I just put some red arrows to make more clear that you have the sudden switch around 1980 and then that faster response later on. And so what we think is happening is that, especially between 1960 and 1980, a lot of iron oxides were coming into these deep basins and were buffering sulfide. So then the question is, how does this happen? Well, essentially, the mechanism is a well-known mechanism for marine geochemists. And that is that if you have a system that has different topography with very shallow areas and deeper areas, you typically see a movement of iron from the shallow areas to the deeper areas, simply because iron can escape from those shallow areas and be deposited in the deeper areas. And we call that iron shuttling. And this concept's very well known. It's been described already in the 90s, and then the model has evolved further. And we also know that it can be enhanced due to eutrophication, because then, as I told you, you get this switch of anaerobic degradation pathways to, from aerobic to anaerobic ones. And so the iron release can be enhanced. And so what we think is happening in this system is that with the onset of low oxygen, iron is being released from shallow areas and is moving towards the deeper areas, buffering the sulfide conditions there. And because these deeper areas are the first one usually to transition towards sulfidic conditions, this is a very important process that can delay the onset of low oxygen. But it doesn't work for much more than a few decades in the system, because at a certain point the system will become more sulfidic, more eutrophic, and you'll get less release of iron, and your iron will be converted to pyrites. And you'll get a deposition of a mixture of iron oxides and pyrites and less buffering of hydrogen sulfide. And this variation in iron source and also the transition in forms is something that is very of great environmental importance. It's something that you then also expect to see in the sediments. So here you could see the iron source, how it varies from oxic sediments to this period with a redox transition when the iron will flux out and be transported, to where the coastal zone is completely sulfidic and there's no iron release. And so what you expect is that if our model is correct, and it's not extra organic matter coming in, but it's essentially an iron buffering mechanism, if that model is correct, we should see this in the iron records in the deep basin sediments. And that is what we see. So this is a record from the same site for which you saw the water column records, and for the same period, where we're using molybdenum as an indicator of anoxic and sulfidic conditions. And so here you can see the iron record showing that there's indeed a rise before 1980 and a drop until 2000. And so we can actually model this and have modeled this, and we can show that also the composition of this iron that's shuttling to this deep area has changed, and you can, or at least you can consistently describe that with the model and can explain sulfide buffering. So here again, we have a mechanism that can buffer sulfide and keep your system from drifting into further low oxygen or sulfidic conditions in this case. So there are two examples here where things get better because of geochemistry. So that is always good news, I think. Now, my third example is on a slightly more negative note, and that is one where conditions get worse because of biogeochemistry. And that is because sediments that are anoxic for a long time, they can start delivering more and more of these reduced components, like methane and ammonium and also sulfide. And this can create a much higher oxygen demand with time, and also you can have a feedback on primary productivity, which makes things worse because then more organic matter comes in, et cetera, et cetera. So this can enhance the intensity of hypoxia and hinder recovery. And as an example, I want to show you the return trajectory. So we saw the trajectory into hypoxia. I'm now going to talk about how easy is it to get a large-scale system like the Baltic Sea to recover from hypoxia. And so we have to look then at the nutrient loads and at the extent of low oxygen in the system. Well, the nutrient loads in the system have been going down for a very long time, almost 40 years now. So since 1980, they're dropping, both for nitrogen and phosphorus. And so you would expect that that would have an impact. But if we look at the hypoxic area, and I've added these red arrows just because I know it's close to lunch, so you need something to make it easier to see the trends. Well, this is, of course, a bit of an oversimplification, but what you do see is from 1900 onwards, the hypoxic area has expanded and really has increased, and actually it still is increasing. Now there's one point though, there is also variation due to physical oceanographic factors like salinity, explaining the dip around 1993, for example. But in the wider context, we can say the nutrient inputs have gone down, the hypoxia has not yet. What's important in this context is that feedback of the nutrients coming in, giving you more primary producers. And in this case, there's a special type of primary producers, and those are cyanobacteria. And cyanobacteria are very well known to fix nitrogen from the atmosphere, so they only need phosphate. And especially phosphate's very, very well available in a hypoxic system like this. So we do have a feedback that also hinders recovery. And just to, especially this year, it's been really bad. So the picture on the left-hand side is a picture from this year showing the massive cyanobacterial blooms in the Baltic Sea, which are in warmer years are worse than ever. Now, getting back to that figure of our trajectories, what you can see is the Baltic Sea. I put this little square where the Baltic Sea is. Nutrient inputs are going down, but the hypoxia recovery has not yet kicked in. And then the big question is, when will that kick in? How long will it take? Is there something we can do to accelerate that apart from reducing nutrient inputs? And could you, for example, induce more burial of that key nutrient for cyanobacteria, and that is phosphate? Could you reoxygenate the sediment and get that phosphate to be buried to iron oxides? See, the iron oxides are playing a central role here. And what we need to do in this case is we need to convert iron oxides we need to convert iron sulfides to iron oxides. So you need to have a mechanism to do that. And we already saw that, at least with reoxygenation, cable bacteria don't seem to be able to do that. But okay, so what have we been doing? So one of the things you can do is you can look at the actual response to natural reoxygenation events that occur in the Baltic Sea. And as I mentioned before, these happen about every 10 years, and they're driven by changes in weather. And changes in weather can lead to conditions where these big masses of oxygenated North Sea water come into these anoxic basins. And the most recent of these inflows occurred in 2014, 2015. And it typically, as you already saw, takes a while for the oxygen to return. In this case, it took a long time. This was a very massive inflow, and it took until, halfway 2016 before it really started changing again. So keep in mind the blue colors here are the sulfide concentrations, green shows there's oxygen. And so what we did was we went to we were actually there by coincidence and took samples in the Baltic Sea and to look at the sediment composition. And what we find is that if you reoxygenate a basin like this, that is so anoxic that you find relatively little iron oxides and phosphorus in the sediment. So there's very little transition of pyrite to iron oxides in this system. And we find that the iron is bound in pyrite, and we do find, and that's an interesting observation, that the phosphorus is associated with manganese oxides, with manganese too. And so these are, as an example, I'm just showing you some micro-XRF results for the surface sediment, where you can see strong enrichments in manganese, you can see enrichments in phosphorus and in calcium. And we see that the phosphorus is in fact associated with rhodochrosite. But the conditions are so reducing in the sediment that you don't form a lot of that either. So there's just a little bit. And you also find manganese 2 phosphates in this sediment. So what we're seeing is that we have this reoxygenation of the system, that we have a lot of oxygen coming in, but these sediments are not starting to take up phosphate. If you look at the water column records, you do see a drop in the phosphate. I'm not showing them here, but the phosphate is actually not going into the sediment as we see here. It's just being pushed to other regions, and so it's staying in the system. So reoxygenation can't do the trick at this moment. And so why is this? Well, essentially this is because of that high upward flux of reductants that is coming from the sediments, and that's removing oxygen, and that's removing any of the other oxidants that can form there and be there in very small amounts. And this is really the legacy of hypoxia that's been reported for other areas, but that really comes to extreme expression, let's say, is extremely visible in the Baltic Sea, much more extreme, I think, than in many other systems. So here it's the methane, especially the sulfide and the ammonium that are fluxing out and that are playing a very important role. Okay, so then the next question is, can the Baltic Sea recover through bottom water reoxygenation? Well, definitely not through this natural reoxygenation. And also because that brings in more salinity that stratifies the system somewhat. So not at this time. And so if you want to reoxygenate the system, you need either a much higher oxygen supply or you need a much lower oxygen demand. And besides eutrophication, that means also that global warming should be addressed, because as I mentioned at the start, global warming leads to change in solubility and also less oxygen supply. So natural reoxygenation will not do the trick. And so if you now look at what we know from models for this recovery, we know that if you stick to dealing with nutrient loads and reduce them further, you can in fact get the system to recover. You'll just have to be very patient, and it can take at least 50 years in the current model simulations. Okay, so the take-home message is here that we have nonlinear trajectories of hypoxia development and recovery in many coastal systems. And I've showed you 3 examples, 2 where you can delay the onset, 1 where bacteria play a critical role, and we really need to understand better where they occur and where they don't occur, because I just showed you 2 examples where they're important, but that might not hold for all of those hundreds of other coastal sites that we see. And also showed you how iron oxide reduction in shallower areas can actually make it more difficult for deeper areas to sink into anoxic and sulfidic conditions. And the final point I made was that if you have feedbacks in the system that sustain the low oxygen, if you have these very reducing sediments, that creates a legacy of hypoxia and can make it very difficult for a system to recover. And in the Baltic Sea, which is the largest system that is affected in this way, we do expect recovery, but it's going to take a while. And so the other take-home message is that we need to understand these microbial and geochemical processes in more detail, because not all these processes are included in the models used for the predictions that I've been mentioning. And if we want to improve these systems or get them to recover, we really need to understand this. And with this, I want to finalize. I want to acknowledge a lot of co-workers of this study, and so their names are on this slide, and I'd like to thank you for your attention.
Thank you very much. So there are 2 microphones in a center row over here. So I would like to welcome questions from the audience. Okay, just walk to the center.
Hi, I guess I'll ask. This is not my major field at all, but I was curious, how much of a sink would you think of carbon are these hypoxic areas? I mean, they're everywhere, and I've never seen anything about how much carbon is actually going into these hypoxic areas. I'm not advocating for hypoxia, but I was just curious.
Well, if you look in Earth's history, of course, these types of systems are important sinks for carbon. And so people are quantifying that. If you look in the modern day environment, well, people are working on quantifying the fjord sink of carbon, which is also, these fjords are also often low in oxygen. So yes, it's important to consider, at least, but it depends on what you're, on what you're comparing it to, if you see my point, the sink of carbon. You know, if you look at CO2 change, it's not an important sink. So.
This is not my field either, but I just start with a comment where, you know, the diagram you show the hypoxia, you know, vary as a function of nutrient loading. But I thought that's very misleading because you reduce the nutrient load, but you are collectively still accumulating nutrients in the marine environment. So that's kind of leading to my question. Do you think it's possible, this is of course a very bold question, is that is it possible to mine the marine sediments for phosphorus? Because we know phosphorus, you know, is much needed for the agriculture, and we are looking for phosphorus as a mineral resources. So what's your thought on that?
Okay, so there are actually two things. I want to comment on what you said about the graph. I don't think it's misleading. I think it's actually the residence time that actually affects the trajectory. So it's true, the larger your system, the slower your response will be. But it is very important to look at the nutrient load because that's ultimately what's driving your system. So the key thing is if you have a large system, it takes longer to adapt. So so that's the point there. The other point about mining sediments for phosphorus, the problem is the concentrations in sediments are relatively low, so you can't really mine that. So you already get problems in the marine environment at relatively low phosphate concentrations. So it's all a question of what you call low and high. but concentrations in sediments are relatively low, so that won't work. So the key solution is to keep the phosphate on land. We should not let it leak into the environment. And what you see is that, especially in the Baltic Sea region, that there's a lot of success in actually keeping there's improvement in keeping the phosphate and nitrogen leaking into the sea, as you could see here. If you look at the phosphorus inputs right now, they're almost in balance with the outputs. At present. So it's only that reservoir that's still in the way.
Hi, I'm Wei Jun Cai from University of Delaware. I really enjoy your talk. Again, another question from people outside the field. Can cable bacteria live in water column at the oxygen-oxygen boundary with hydrogen sulfide and oxygen mixing there?
Sorry, can you?
Can cable bacteria live in water column with oxygen and hydrogen sulfide at the oxygen-oxygen boundary and play a geochemical role there?
Oh, if they can live in the water column. I don't think so. Yeah, what people have found is that they can stick, or at least there are reports that they might, stick their head out of the sediment. So they might, sit in sulfidic sediments and then grab the oxygen from the overlying water. But I do think they need some type of fixed space. But this is something. Yeah.
So if a system with stable hydrogen sulfide in the bottom, and oxygen right above that. But anyway, nobody ever found that.
Yeah. Okay. Yeah, so what we see is in a lot of systems, we see sort of like a boom-bust cycle. So they, if you try to have them in a lab incubation, they grow fast, and at a certain point, they stop. And so, yeah, that's a bit outside of what I should talk about here today. But so there's still a lot to be found out about those dynamics.
One last question, ma'am. So, yeah, just one last question, ma'am. Maybe I'm running out of time. Just, the Disulfo-Bulbaceae family, they are also known for their sulfate reduction. Yeah. So what is the control or what is the switch that they really employ so as to choose between their electrode acceptors, like when to really go for oxygen and when to go for sulfate reduction? Can you elucidate something on this?
No.
Because
no, so I can't, yeah. I'm not a microbiologist, so I won't comment.
Yeah,
on
basically
this.
when
I think one of the big questions actually is we know there's competition. You could see that in the data that I showed. There's competition also with other sulfur oxidizers. And so there's competition with Beggiatoa for the same niche. And in this Dutch lake, they just vary over the season. We don't really know why. And in the Baltic Sea, in these reoxygenated areas, we find Beggiatoa, and we don't find cable bacteria, or at least in low abundances, they're there. But the Beggiatoa win there. And so I didn't really touch upon this here, but there is competition, and we need to understand that to understand their environmental relevance in the hypoxic zones in other systems. Yeah.
Thank you. Thank you. Take one more question.
Carolyn, thank you for a really lovely talk. This is more an observation. I was really intrigued when you showed how long it takes to essentially reoxidize that sediment, that there's implications for the deep earth and the evolution of the great oxidation event that's attached to this. The idea that it takes a long time to titrate out all of that reduced species, even as you start to add oxygen back to the system. And so I was really intrigued by the connections between your modern study and the potential implications for the early Earth and the evolution of oxygen in the system. Thank you.
Yeah, I agree with what you said. Yeah, thanks.
All right, I think, all right, so let's thank Caroline another time, and let's conclude this plenary session.